Hello everyone. Welcome to the new session of Organization of Genetic Material RNA. So in the last session we have studied about the DNA. In this session we are going to study about the structure of RNA, right? So ribonucleic acid is a molecule that is present in the majority of living organism and viruses, right? So they are present in majority of living organism. So they are present in prokaryotes as well as eukaryotes, right? Eukaryotes and some viruses also. Viruses having their genetic material as RNA. Okay, so one of this is HIV virus, right? Okay, so this is present in the case of prokaryote. It is present in the form of rRNA, and also tRNA and mRNA. Three different forms of RNA are there. That is rRNA, tRNA and mRNA. In the case of eukaryotes also, it is present in the same form, right? It is made up of nucleotides. So in the last session, we know about what nucleotide is made up of, and it is the building block of the genetic material, right? So nucleotide is made up of sugar plus nitrogenous base and phosphate group, right? So these three things, sugar, nitrogenous base and phosphate group, join together to form nucleotides. That forms nucleotides. Complex compound of high molecular weight. So RNA are complex compound, okay, of high molecular weight that function in cellular protein synthesis. So with the help of RNA, different proteins are synthesized in the cell. Okay, so there are three different types of ribosomal. Okay, so structure of RNA. Now we will discuss about the types of RNA. So we are having three different types of RNA as rRNA, that is ribosomal RNA. This RNA is present in RNA, so they are named as ribosomal RNA. tRNA, they are present in transfer RNA. And mRNA, they are present in messenger RNA. So this R, T and M for ribosomal, transfer and messenger RNA. So we will study one by one what are the structure and what are their functions of these three different types of RNA. Okay, so ribosomal RNA is the most abundant RNA, that is it constitutes about 70 to 80% of the total RNA present in the cell. Okay, and this is 3 to 4 types. There are 3 to 4 types of rRNA. So how many types of RNA we are having? 3 to 4 types of rRNA, right? And this is the most abundant RNA present in the cell, that is 70 to 80%. Very important to remember. Okay, so RNAs of the eukaryotes are 4 types. Now we are having 4 types of RNA, 4 types of rRNA in case of eukaryote. What are those? 28S, 18S, 5.8S and 5s. So here it is eukaryotic RNA. So if you see the figure, this eukaryotic RNA, so it is having 18S rRNA, 30 proteins, okay, and they are having 28S, 5.8S and 5S. So if you just see the rRNA part, that is 18S type, right? First one, 28S type, 5.8S and 5S. So four different types of RNA are present in the case of eukaryote. So 18S is present in the 40S, that is the smaller subunit of RNA, smaller subunit of rRNA, right? And 28S, 5.8S and 5S, they are present in the larger subunit of RNA, larger subunit of rRNA, that is 60S type. Okay, so if we talk about the prokaryotic ribosome, then we are having three types. Okay, what are those? 23S, 16S and 5S. So they are having, if you see the smaller subunit of prokaryotic RNA, then they are having 16S small rRNA, and they are having 23 and 5S type in the larger subunit, right? So in the 30S, that is a smaller subunit of prokaryotic RNA, they are having 16S rRNA, and in the larger subunit of the prokaryotic RNA, we are having 23S and 5S type of RNA. So here we can see that this ribosomal RNA means ribosomes are made up of these different types of rRNA. And in the case of eukaryote, we are having four different types, and in the case of prokaryotes, there are three different types. So very, very important point to remember, right? Moving forward, so 28S, 5.8S and 5S occur in the larger subunit of ribosome, while 18 is found in the smaller subunit of ribosome in the case of prokaryotes, right? In the case of prokaryotes, rRNA is transcribed in the form of longer chain, 45S in the case of eukaryotes and 30S in the case of prokaryotes. So rRNA, when it is transcribed, means from the DNA, this RNA gets synthesized, right? By the process of transcription, right? And in the case of eukaryotes, right, this RNA is of 45S type. Okay, later what happened? Splicing of this big part, that is the 45S RNA, takes place, and smaller subunits are synthesized, right? That is of 23S, 5S, 21S and 16S, like this, right? So four different types of these ribosomal RNA, okay, are synthesized from the transcript of 45S type, right? And in the same way, this 30S in case of prokaryote, the transcript is made up of 30S type, and later with the help of a splicing process, it is broken down into three different types, that is 18S, 16S, 23S and 5S type. Okay, this 16S, 23S and 5S type. Whereas in the case of it was 18S, 28S, 5.8S and 5S. Right? So this is occur in the case of eukaryotes and prokaryotes. 30s subunits of the prokaryotic ribosomes has 16s ribosomal RNA and about 21 protein molecules. So smaller subunit that in the case of prokaryote, that is 30s subunit type, and it is having 16s rRNA, 16s rRNA, which is made up of about 21 protein. Along with the 16s RNA, they are having 21 protein molecules also. 50s subunit of the prokaryotic ribosome, it is having 23s 5s type of rRNA, right? These are the two different types of rRNA are present in the 50s type of subunit of the prokaryotic ribosome, along with 34 protein molecule. Okay. In the same way, the larger subunit in the case of eukaryotic ribosome, that is 60s type, it is having 3 different types of rRNA, that is 28s rRNA, 5s rRNA, 5.8s rRNA. Okay. 5.8s rRNA and about 40 protein molecules. 40 protein molecules. Okay. Okay, coming to the smaller subunit in the case of eukaryotic ribosome, it consists of 18s rRNA and some 30 protein molecules, are present. Okay. So here we can see that the ribosome, whether it is in the case of prokaryotes or eukaryote, it is made up of two components, two biomolecules, that is rRNA and proteins. Okay. Okay, the second type of RNA, that is the transfer RNA. It is also called as the soluble or sRNA. Okay, so there are over 100 types of tRNA. We are having different 100 types of tRNA. So we have just now seen that rRNA is of 3 to 4 different types, but tRNA is of 100 types. Okay. Transfer RNA constitute about 15% of the total RNA, 15% of the total RNA, and it is the smallest RNA. Okay. Most abundant type of RNA is the ribosomal RNA, and that percentage is about 80% of this total RNA present in the cell. But this transfer RNA, it constitutes just 15%, right? Okay. So tRNA is the smallest RNA with 70 to 85 nucleotides long with the sedimentation coefficient of 4s. So its sedimentation coefficient is smaller than the smallest rRNA, that is 5s type, which is present in the ribosome, Okay, so here the sedimentation coefficient of the tRNA is of 4s type. So the 5 regions are unpaired or the single-stranded AA binding sites. So we are having 5 different regions in the case of tRNA. So we are having T5C loop, DHU loop, extra arm, anticodon loop, and the acceptor RNA. So here we can see this is the DHU arm, this is the T5C loop, right? This is the anticodon loop, very important. And here we are having acceptor arm, this is called as acceptor arm. Okay, and we are having the extra arm or variable arm. So this is the tRNA 2-dimensional structure, and we are also having the tRNA 3-dimensional structure, as we can see here, right? Okay. So first one, the anticodon loop. So it is made up of 3 nitrogenous bases of the recognizing and attaching the codon of mRNA. So we can see here we are having this codon on the mRNA. This is the mRNA, right? And its just complementary anticodon is present here. GAG it is CUG. Anticodon is CUG, right? And that is why it is called as anticodon loop. Now this CUC represent the glutamic acid. So according to the anticodon present on the mRNA, we are having a specific anticodon on the tRNA in the anticodon loop, right? And this tRNA, this specific tRNA is having the glutamic acid attached to it. Here CUC is for the glutamic acid. So as this anticodon changes, the anticodon also get changed with a specific amino acid attached to the acceptor arm, right? So this anticodon is very, very important. Acceptor arm or AA binding site. Acceptor arm or AA binding site. It lies at the 3' end opposite to the anticodon and has CCAOH group. CCAOH group. So here we can see that at is 3' end. 3' end acceptor arm is there and anticodon has CCAOH group. CCAOH group. This CCA group is added after the transcription. So this is a modification at the acceptor arm. This is the modification at the acceptor arm, that is addition of the CCA group, right? And with this group, a amino acid is attached, right? So amino acid or the AA binding site and the anticodon are the two recognition sites. So this particular acceptor arm and this anticodon are the two recognition sites of the tRNA. This is very important, and question can be asked on this. So how many types of recognition sites are present on the tRNA? So two recognition sites, one in the anticodon and one at the acceptor arm. Okay. T5C loop. So here we can see the T5C loop and it contains a pseudo-uridine. So pseudo-uridine is the modification of the uridine nucleotides and the loop is the site for the attaching to the ribosome. So this particular loop helps tRNA to get attached to ribosome, when during translation process, that is the protein synthesis process. So this particular loop is having its significance that it helps tRNA to get bind to the ribosome at the time of translation or the protein synthesis. Extra arm. It is a variable side arm or the loop which lies between the T5C loop and the anticodon. So here we can see the variable arm or the extra arm. It is present between the T5C loop and the anticodon. It is present in between these two arms, right? The exact role of the extra arm is not known, but it is variable in the different types of tRNA. Okay, so that is why it has got its name, variable or the extra arm. DHU loop. The loop contains dihydrouridine. Again, it is a modified uridine nucleotides, dihydrouridine. It is a binding site for the amino acyl synthetase enzyme, right? So this is very, very important for the translation process. Amino acyl synthetase enzyme, this is the site for the binding of it, right? tRNA carries specific amino acid at a particular point during the polypeptide synthesis as per the codons of mRNA. So this tRNA, every tRNA is having the specific amino acid at a particular point during the polypeptide synthesis. So according to the codons present on mRNA, we are having a specific tRNA, right, having attached amino acid according to the codon present on the mRNA, right? So the codons are the recognized by the anticodons of the tRNA, right? So this codon which is present on the mRNA are recognized by the anticodon loop present on the tRNA. Very, very important point, right? So the specific amino acids are recognized by the particular activating or the amino acyl synthetase enzyme. So this specific amino acid are recognized by activating or the amino acyl synthetase enzyme, right? The third type of RNA, that is the messenger RNA. Messenger RNA. It is a long RNA which constitute about 2 to 5% of the total RNA. So this is the least amount of the RNA. This type of RNA is the least amount of RNA present in the cell, right? It brings instructions from the DNA for the formation of the particular type of polypeptide. So inside the nucleus, DNA is present, and on the template of the DNA, RNA are formed, and it comes from the nuclear pore. Nuclear pore, this mRNA comes out of this, right? From the nucleus it comes out, and this get modified as it gets synthesized. How? We can see here the capping, we can see here the poly A tail. So this is very, very important. What is the function of it? The instructions are present in the base sequence of its nucleotides, right? And it is called as the genetic code. All the instruction which are present on the DNA is passed to the mRNA, right? So 3 adjacent nitrogenous bases specify a particular amino acid. Formation of polypeptide occur over the ribosome. So this mRNA is very important. It is having this transcript. It is having a capping here, a cap, right? A cap is attached, and a poly A tail at 3 prime, and poly A tail, this is called as poly A tail. This is for the protection, because many nuclease enzymes are present in the nucleus, they can digest this. So what happened? The capping is done as get it synthesized. The capping is done at the 5 prime end, and at the 3 prime end poly A tail gets synthesized to just protect this mRNA transcript, right? And on this they are having all the codons, right? That they are having all the codons, and it helps to synthesize the specific protein, right? So this is all about this session. We completed the structure of RNA in this particular session. Okay, we will meet with one more interesting session. Till then, thank you.